Hello class, this is Mr. Mormon. Welcome to our first video on genetics. This will be an introduction to our genetics unit. So let's start with DNA. You can't talk about genetics without first understanding DNA. Okay, so just as a quick review, DNA is deoxyribonucleic acid. It's the blueprint of life. It contains the instructions for making proteins. This diagram explains it a little further. Inside a cell in your body, actually in every cell in your body, is a nucleus. And inside that nucleus you will find chromosomes. Chromosomes are made of DNA. Now remember that DNA and the chromosomes, they always come in pairs. One half of the pair has come from the mother and one half has come from the father. In humans, there's 46 total chromosomes, which come in 23 pairs. There's 22 autosomal chromosomes. Those are the ones numbered 1 through 22. The largest chromosome would be number 1. The smallest would be chromosome number 22. They're in order of size. And then there's the sex chromosome. The last pair is either XX for female or XY for male. Recall also that the information that DNA stores is in the rungs of the DNA ladder. There's 4 different nitrogenous bases, A, T, C, and G. A always combines with T in the ladder, and C always combines with G. They're called complementary base pairs. But A, T, C, and G, adenine, thymine, cytosine, and guanine, are the 4 letters of the DNA alphabet, which spell out all the information to make all the different proteins. Now, a gene is a segment of DNA with instructions to build a complete protein. So this might represent a gene right here. This segment of DNA could have information to make a complete protein. Maybe this is a protein that makes the pigment color of your skin. Maybe this is a protein that makes your hair curly. Remember also that proteins could be enzymes which are controlling chemical reactions. So all of these instructions come from the DNA. So that's a... Now, DNA makes proteins. What you need to understand is that basically we are made out of protein. Our skin, our hair, our bones, all the different parts of our body are made of proteins. And it's the DNA that has the instructions that make all these proteins. There's proteins that make the color, the pigment in our skin. There's proteins that make the color of our hair. All of this is controlled by DNA. The traits that we can see in these people, the physical characteristics, are all being controlled by their DNA. Now we need to know the vocabulary words phenotype and genotype. Phenotypes are all the traits that we can see in these people. Genotype are the genes that created those traits. You cannot see genes. You can only see the traits that are being created by those genes. The thing to realize here is that all of these traits, being able to curl your tongue or not being able to curl your tongue, a widow's peak, attached earlobes, unattached earlobes, free earlobes, hitchhiker's thumb, hair between your finger joints, a cleft chin, freckles, these are all traits that are controlled by DNA. It's your DNA that's making the proteins that gives you these traits or characteristics that you can see. So the genotype is what gives you the instructions for the phenotype. Now, heredity or inheritance is the passing of traits from parents to offspring. Genetics is the scientific study of heredity. Gregor Mendel is considered to be the father of modern-day genetics. As a matter of fact, it's often referred to as Mendelian genetics. Gregor Mendel was a monk, and part of his job was tending to the gardens at the monastery. Mendel noticed that many of the plants he worked with came in different varieties. In this picture, we see Mendel tending to his pea plants, which is one of the plants that he did a lot of his studies on. This diagram shows some of the great variety that Mendel saw in the different pea plants. Purple and white flowers, different positions of the flowers on the stems, colors of the peas and pods, wrinkled or pinched pods or seeds. Now, what Mendel knew was that if he bred plants with the same traits or characteristics over and over and over again, like tall with tall, tall with tall, tall with tall, over and over and over again, what he ended up with was a purebred strain where he would always get tall when he would mix them. And when he bred short with short, he could create a purebred strain. So taking the same traits and breeding them over and over and over again would create a purebred strain. Once he had purebred strains of, let's say, tall and short, he then tried crossing them, crossing the purebred tall with the purebred short. What he got was surprising. He expected to maybe get something in between, but what he got in each one of these examples where you see he crossed the purebreds of the opposite strains, he ended up one showed up and the other disappeared. Okay, 100% of the time when he crossed tall with short, he got tall plants. This was very confusing to Mendel. So alleles are the different forms of a gene. A purebred is going to have two of the same alleles. A hybrid or heterozygous are going to have two different alleles. Now, homozygous and purebred mean the same thing, two of the same alleles. Hybrid and heterozygous mean the same thing, having two different alleles. So what you need to understand is that Mendel was controlling the breeding of the plants. He was deciding which plant was going to be the mother and which plant was going to be the father. Now, to understand how this works, you need to understand that plants are actually reproducing sexually. Inside the carpel here are little ovules. They're basically eggs. But in order for them to develop into these peas or seeds here, they have to be fertilized first. And that's the job of the pollen. Pollen contains sperm cells, and pollen is spread by bees or it's blown through the air, and it pollinates the flower, and those eggs become fertilized and can then develop into the seed or and the fruit around it. If pollination never happens, you never get seeds or fruit. So Mendel was just controlling all this. So in this example, he's choosing this plant to be the mother. So he cuts away the stamen, which are what are producing the pollen. These are the stamen around the outside of the carpel. So he cut them away here. You can see there are no stamens. So this plant can now not fertilize itself. What he then would pick the plant that he wanted to be the father, and he would play the job of the bee here and would collect the pollen and transfer it to the carpel, fertilizing the ovum, the eggs inside the carpel, and it would then develop into a piece. So these seeds now, he knows that this purple plant was the mother and this white plant, white flower, was the father. So he was controlling which plants were breeding with which plants. Okay, so now back to his tall plant, short plant cross. This is the parental generation, or the P generation, purebred tall crossed with purebred short. What happened in the F1 generation, or first filial generation, is no matter how many times he did this cross, he always got tall in this next generation. The shortness trait seems to disappear. If you breed the F1 offspring, you get 3/4 tall, 1/4 short. That short trait shows up again. It skipped a generation. Now, Mendel wasn't the first to notice this kind of thing, but he was the first person to figure it out and explain why it was happening. What Mendel figured out was that there were different alleles or factors, different forms of the genes. Here we see tall and short. Big T is the allele for tall. Little t is the allele for short. Here we had a purebred tall crossed with a purebred short. In this next generation, no matter how many times you did it, the tall plant would always give a big T. The short plant would always give a little t. And these all ended up as hybrids. Now, hybrids look tall because the tall trait is dominant. These actually have both a tall and a short allele, but they look tall. If you then breed the ones in this generation, hybrid with hybrid, you end up with the combination that you get down here. 1 out of 4 will be purebred tall, 2 out of 4 will be hybrid tall, and then that short trait shows up again in about 1/4 of the offspring. In this chart, you can see that you get that same 3-to-1 ratio When you cross any of these purebred dominant with purebred recessives, and then you do it for 2 generations. So the F2 generation, you get that 3-to-1 ratio again. 75% will have the dominant, and then the recessive trait shows up in 25%. Okay, so remember, the dominant allele is always going to show up, whether it's purebred for the dominant or if it's a hybrid. If the dominant allele is present, you are going to see that trait. The recessive alleles can be covered up or masked by the dominant alleles. And I just want to finish up with a little bit about the notation in genetics. Remember that the dominant allele always picks the letter. The recessive allele gets a small case letter of the same thing. Here's what I mean. In Mendel's pea plants, tall was dominant over short, so we use a big T to represent the allele for tall. We do not use an S for short, we use a little t. The gene is the gene for height. The alleles are tall and short. Big T for tall, little t for short. Okay, that's about enough for this video. Again, if you have any questions, please ask me tomorrow in class.